Hey guys, it's Standing for Truth coming to you tonight. I know it's a little bit late, but I got some good friends here. I've got Nephilimfree here and Jeremy and a few other people I think that'll be joining a little bit later. We're going to be talking about creation, evolution, just kind of having a free flowing dialogue, have some fun, talk about hopefully some evidence against. Pawns come to people evolution. The first thing I wanted to do is introduce Jeremy to the live stream. Jeremy, if you want to say a few words, and then Neff as well, you can say a few words as well, and we'll go from there.
I'll speak first.
Yeah, you can go ahead, Jeremy.
Yeah, I'm Jeremy. I live in Southeast Asia, Philippines. Um, I'm a former atheist. Uh, I, I've been studying the creation evolution thing for about 15 years and counting. Um, and I think that's it for now. Thank you. Hello?
I think I'm just gonna mute Jeremy real quick. I think he's got a. Is that his echo that you hear there, Neff? Or is that? Oh. Okay, we figured it out. Neff, if you wanted to say a few words, I'm just gonna unmute. Actually, Jeremy, you're just muted right now. If you wanted to unmute yourself real quick, brother. Athena, can you hear me?
Yes.
Neff, can you hear me? And Jeremy, can you, are you there?
Yes, I'm here.
Okay, perfect. And Neff, are you there, brother?
Yep, I'm here.
Okay, perfect. So it looks like we got Athena and Crispr here as well. So thanks for joining us, guys. I'm not sure if you've dialogued. I know you wouldn't have dialogued with Jeremy. This is his first time, but I assume you've probably dialogued with Neff here a time or two.
Yeah, I think I've talked to Neff once or twice.
Great.
Yeah, I believe
Good. Neff, I wanted to start off with our discussion with RJ Downard and Jackson Wheat last week. They seem to have a problem with the points we were bringing up in relation to the 90% of species origin, the article, the technical paper. I know you did a bit of a live stream a couple days later explaining why your line of questioning made sense and was necessary and kind of getting, to the bottom of it.
right.
Can you kind of explain that a little?
Well, what I was talking about was the scientific studies. There's actually another one that mirrors it, but I was talking about a scientific study that made an argument that the vast majority of single nucleotide changes to the DNA of the human genome have taken place in the last 5,000 to 10,000 years. And I gave an argument as to why that's implausible if evolution is true. Humankind has been around for hundreds of thousands of years. And the people that were in the hangout with me just went ballistic on me. I tried to set the ground for the understanding of the argument, and then asked a question, and it didn't seem like anybody wanted to answer the question. But it's basically a two-part argument. It's part of it is that evolution can't be true because of this evidence, and the time span, the uniformitarianist evolution / evolutionist time span for the history of mankind, in fact, all biological life on the planet, is an outrageous extrapolation, a stretching of the past. And so that's what I was talking about.
And then do you, I'm not sure if you listened to that there, Crispr or Athena, but do you have an objection to what Neff was putting forth in regards to that?
Well, I didn't hear the original discussion, so I don't actually know what it is that he's putting forward.
Neff, what would you say you're putting forward that supports our view of the biblical model as compared to, an evolutionary long time frame model?
Well, the point of the argument is that if the vast majority of single nucleotide changes occurred to the human genome in the last 5 to 10,000 years, that's a refutation of evolution, because if mankind had been around for hundreds of thousands of years, single nucleotide changes to DNA would have been an ongoing thing. In fact, it would have been the very mechanism that created a large portion of the DNA molecule and much of its information, almost all of its information, in fact. So that it's not logical that if man has been around for hundreds of thousands of years, that the vast majority of changes to his DNA by single nucleotide changes, that's a single nucleotide, a missense mutation, point mutation, that changes just one little bit of data in the DNA, that should have been an ongoing process, and it should not be that the vast majority of those changes occurred in our DNA in only the last 5 to 10,000 years. So it doesn't seem to add up for evolution.
Do you have specific references on that?
Yeah, I'll be happy to provide one. Let me see here.
Oh, it looks like, hey, Pure Facts, how you doing, brother? Can you hear us? I think Pure Facts, can you hear us? Looks like you just joined the group. Okay, well, whenever he figures it out there, but in regards to references, if that's
here it is, you should be able to see it on the screen now. Is it showing up?
Can you see that, Crispr?
Should be in the Hangout on the screen.
Yeah, just a second. Let me type in that team ID.
Okay.
Let's see here.
You can stop sharing it. I got the PubMed ID out of it.
You got it?
Okay.
Yeah, so I mean, just reading the abstract here, actually what they say is that they're estimating that inside the single nucleotide variants within protein coding regions that are predicted to be deleterious arose within the last 5 to 10,000 years. So that means the ones that have a demonstrable harmful impact and would presumably be selected against are all very young, which is actually expected under an evolutionary model.
Well, the problem, though, is that the vast majority of single nucleotide polymorphs are deleterious. Virtually all of them.
Not true.
Yes, it is.
There's something like 1.5 million SNPs. No, it's more than that. It's much more than that. And most of them are not strongly associated with any disease.
No, well, the vast majority of SNPs are deleterious. I'll provide sources for that.
Yeah, I mean, I'd be interested to see what sources you could provide, but that's simply not consistent with the current literature.
Well, Crispr, if we see that, I think we agreed to it before that, human mutation rate is, say, 100 mutations per person per generation, indicating that our children are going to have about 100 mutations more than, say, we have. And then that means our grandchildren are going to have 100 more mutations than they have. On a population level, isn't that destructive? And how is mutations / natural selection going to build the genome in order for something like, say, your pond scum to people evolution to be even considered empirical observational science?
So what specifically are you asking me right now? Because that sounded like it was actually several different questions, and I want to try and go one at a time.
I think I'm just elaborating on what Neff here is saying in regards to the fact that mutations, human mutations, seems to be catastrophic, detrimental, and deleterious. So how is that going to build the genome? And in regards to what he's saying about the SNPs, where's the resolution to that in a evolutionary, say, model and worldview? And if you can expand on that a bit more, Neff, just to make a little more sense to CRISPR there.
Well, I mean, I would say that the first thing is that, the majority of mutations are not going to actually be harmful in any way. That's what the current evidence is. Now, I understand that you guys might not agree with that, but that's what the evidence is. So, this idea that all of these mutations are going to be necessarily and intrinsically damaging is simply not supported in the literature. To the question of how do you get how do you get actual development from that? That's obviously a somewhat more complicated question to answer, but you can look at the range of mutations that are observed, and you can see that some percentage of those mutations have impacts on morphology, which are then heritable. And obviously, any impact on morphology can be either beneficial or detrimental, depending on the specific nature of the environment that organism is in, right? So, if the particular change in morphology is again, these changes tend to be very slight. They tend to be percentage based. They tend to be these are minor modifications by and large. These are not things like the duplication of legs onto where the antenna go, for instance. These are, which is a large scale change in morphology. These are minor changes, and minor changes in morphology can either be beneficial or detrimental. The same change, that is, depending on the precise nature of the environment that organism is in. So yeah, that and then you can see how the accumulation of such things would lead to differences between organisms. But again, I don't think that's really the question you're specifically asking. So, well, I guess it is, right? Because that's saying how do you get the varieties from mutations? Well, that would be an answer, but
I would have to say that I think it's an atheist myth that the majority of mutations are not deleterious, that they are absorbed by, this is how the junk DNA paradigm came into being. The idea was that the deleterious, it goes all the way back to Haldane's dilemma in the 1930s. This idea that the vast majority of the DNA is junk and will therefore absorb the vast majority of mutations. And the protein codes, when they receive a mutation, natural selection is going to fix the problem. But numerous studies have been put forth that keep coming up with the same problem. In fact, one by a famous Japanese researcher in 1968 said, in order to combat the deleterious effects of random mutation, people would have to have 44,000 offspring each set of parents, 44,000 children, and only 2 of them survive. That's how bad it is.
Yeah, so I actually know exactly the paper you're referring to. That particular paper ignored beneficial mutations, and it ignored beneficial mutations because including any level of beneficial mutation at all had the rate of evolution far exceed observation, which just suggests that the model that they were using isn't perfect. But basically, if you want to use that paper, you're already refuting yourself. So I would not recommend that.
I don't see how. He's not the only one. There's a number of scientists that come to the same conclusion, some atheists and some Christian creationists. But this idea that these so-called beneficial mutations, we just don't know of them. Recent studies in genetics have said, quite a few have said, in fact, some of the researchers that we're talking about have said, the rate of beneficial mutations is so slight that it's only perceivable in population genetics. Can't hardly even be counted. So I don't know. I know the evolutionists love to believe. I think this is just another atheist myth that there's all these beneficial mutations and nobody can seem to point out any that have any.
I can answer that immediately.
Well, nobody can seem to point out any that have any effect other than something like gaining an immunity to a drug, which has no effect, no potential to move an organism towards something other than what it is.
A simple one? There's no mutation in humans. I can pull it up if you give me a few minutes. But basically, it affects the length of the long bones of the leg, so you end up with a slightly longer femur.
Okay, so
this is intrinsically neither beneficial nor detrimental. However, in certain circumstances, in certain environments, it would be beneficial, and in other circumstances and environments, it would be detrimental. In other words, when you observe a single mutation, you're unlikely to observe it as being beneficial. It's only when the environment makes it beneficial, right? That's what matters, is the change in the environment.
Okay, so how does that affect the design of the organism? Longer bone, how does that move it towards becoming something other than what it is? The organism already has bones.
No organism ever becomes anything other than what its ancestors were.
That's what evolution claims.
That's absolutely not what evolution claims.
No, evolution claims fish begat something that was not fish.
Nope.
You don't understand.
If you're defining fish in a scientific manner, it has to be monophyletic.
Okay, so I don't understand what you're saying here. You do understand that evolution posits that over multiple generations, the offspring of fish were land-dwelling creatures with lungs and legs and digits and skin instead of scales, right?
It posits that Sarcopterygians.
Wait, do you understand what I just said?
Let me rephrase what you said using scientific language.
No, I just want an answer to my question.
Yes, I understand what you said. Let me explain what you said using actual scientific language. Can you mute the guy who's got all the background noise?
I just muted Jeremy. I think he's the one with it.
Yeah, no, I think he was. So it posits that there were Sarcopterygians that gave rise to tetrapodal Sarcopterygians, that gave rise to amniotic tetrapodal Sarcopterygians. They're all still Sarcopterygians.
Okay, so applying a title doesn't change the fact that fish ultimately begat land-dwelling creatures with a different anatomy.
Can you name?
That's what evolution claims, is it not?
Can you name for me?
I just really want to answer.
Let me ask you a question. It's a very simple question.
No. I asked a question. You're really trying hard not to.
I asked a question. You asked a question, and I answered it specifically.
No, you didn't. You used the title Sarcopterians as though this
Sarcopterygian.
in order to not directly answer the question, isn't it true that fish over multiple generations produced creatures, land-dwelling creatures with different anatomies that are not fish?
They are still everything their
You can't say
ancestors
yes,
were.
can you? You just can't do it.
Because fish is not a scientific term.
Oh, okay, oh boy, we're going down this road now.
Is a cuttlefish a fish? Because people call it a fish because it
Okay.
swims in the ocean.
Oh, okay. So primates came into being from a non-primate, right?
Yes, that's true.
Okay, so fish, whatever you want to call them, ichthyosteids, whatever kind of a thing.
Sarcopterygians.
I don't care how you classify them. Classifying and throwing a name out is not really answering or directly answering the question. Okay, evolution posits new features arise, ultimately resulting in different body plans, different anatomies, producing different types of living things
Let me
that are
say
not the same thing as the thing that they came from. That's just, everybody knows
this
this.
way. I think it'll make you happy.
So
There's a group.
the point I'm asking,
Let me answer your question.
back to my original question.
Talking and let me answer.
My original question.
Stop talking and let me answer.
No, my original
Stop talking
question
and let me answer.
How does the
Stop
thing that
talking
you
and let
put
me
forward?
answer. Stop
how does
talking and let me
How
answer.
does the thing that you
Stop
put
talking
forward
and let me answer.
move the organism
Stop
towards
talking and let
becoming?
me answer.
Okay, if you want to
You'll
answer that
get your
question,
answer if you stop talking.
no, you don't even know the question
You
anymore.
have to stop talking.
Okay, guys, what we'll do? Okay, what we'll do is we'll let Neff ask the question clearly, soundly, and then we'll let Crispr take as much time as he needs to respond to the question, and then we'll go from there. Okay, go ahead, Neff. Ask your question, and then we'll allow Crispr to respond.
Okay, so the original question, which you went all the way around, was I asked, how is this mutation of getting a longer thigh bone move the organism towards becoming a different type of organism than it is when it's not an arisal of a new feature? The organism already has thigh bones, you see.
Okay, go ahead there, Crispr.
Right. And so that entire question is irrelevant because evolution does not suggest that organisms become something other than what their ancestors were.
Oh.
Now, if you want to explain, if your question is how do morphological changes lead to the development of features different than what the ancestors had, that's a different question. I can answer that too, but I think he's
So
I can answer that question.
So you don't believe that evolution theory posits that different types of organisms arise because of evolution from their antecedent, right? The anatomies don't change, and elephants didn't come into being from something that was a non-elephant, and whales didn't come into existence from a land-dwelling creature. None of that's true. That's not what evolution says.
They carry forward the traits.
But is that not true? Yes, but is that not true? That result of that.
There is descent with modification.
I understand you believe that.
What is being modified are the existing characteristics that are carried forward with modification.
We all understand that's the theory. I'm just asking you, don't you do accept, no you're not, that a different type of organism comes into existence when it did not exist before, from a different type of organism. That's what evolution is. So my question was, how does getting a longer thigh bone move that organism towards, in the process of becoming, producing a creature with a different anatomy? So we can see evolution produced land-dwelling creatures, produced fish, produced land-dwelling creatures, Tiktaalik rosea produced ichthyosteiga.
Sure.
You understand.
Sure.
Right? Can you answer?
If you look at the basal sarcopterygians, they already have all of the bones that the tetrapods have. So you don't need any new bones to get a tetrapod from a basal sarcopterygian. What you do need is changes in relative size and proportion, which is exactly what this mutation that I've referenced does, is that it causes a change in relative size and proportion.
Okay, so, but new features don't arise by changing the proportion and size of extant ones. See?
Well, is the limb of a tetrapod a new feature compared to the fin of a sarcopterygian?
Well, digits come into existence that didn't exist before. Lungs came
into
existence.
What's a digit made out of? What's the components of the digit?
New bones, more bones than existed before.
No, those bones were actually already present in the Sarcopterygian.
Not in the fish.
In the Sarcopterygian,
Not in the fish.
they were there.
They weren't there.
You had phalanges.
Listen.
You had phalange homologs in the Sarcopterygian.
They're made of keratin in the fish, not bone. The same stuff your fingernails made of. Okay, not bone.
Yeah, you have to go past that, and you see where the actual bone is. It's
right.
It's what you have just past the radii, right? That is the phalangeal homologue.
So new bones come into existence during this process.
They're not new bones.
I just
all
told
the
you
bones required. Yes, you said something that is incorrect.
In the fish, they're not bones. They're keratin in the lobe-finned fish. Okay, they're not bones.
Yes, the lobe-finned fish had more features than the tetrapod does.
So I say the bones came into existence for the digits in land-dwelling amphibian tetrapod.
Yes, those bones
don't exist in the
lobe-finned fish. For the tetrapod,
they don't exist in the lobe-finned fish.
Yes, you're just simply mistaken.
They don't exist in the lobe-finned fish. That evolution supposedly was the evolutionary ancestor of the amphibian tetrapod. I'm sorry, that's not so.
The homologous structures already existed, and they developed from the exact same developmental pathways, from the same structures, in the same order, in the same places.
The fish doesn't have the bones that there exist as digits in the amphibian tetrapod.
Sharks don't have any bones, but they have structures that are homologous in all of the same places that develop from the same underlying structures.
But that's not the case with lobe-finned fish.
Do you know how much of a change that requires to actually have calcification of the structure? It's not much.
I'm just trying to help you understand. The bones are not passed on. They didn't come from the fish. They're not present in the fish. New bones had to arise. The structures are all present. No.
Yes, I know that you're mistaken about this, but that's not my fault. That's because you're not familiar with the literature.
No, it's not so. I debated Per Alberg, an anthropologist at the University of Uppsala, about this, and he admitted that bones have to come into existence where they don't exist in the fish. I don't know why you're not up with that. But so
because I've actually seen structural diagrams,
and then lungs come into existence when they don't.
Oh yes, let's talk about lungs too, because they already had lungs.
Let's slow down a little bit. Let's go back to the premise. My question. Okay.
No, actually, I want to talk about lungs. I think that's a great topic. Let's talk about lungs.
Way around the question. The question was, how does getting a longer thigh bone move the organism towards becoming a different type of organism with a different anatomy, because that's what evolution posits.
Well,
you have not answered
it has a different anatomy because it has a longer thigh bone.
No, that's not a difference in anatomy. That's a difference in morphology. That's form and structure, not anatomy and body.
Yeah, well, if you keep pushing that out, eventually you got to call it a difference.
Well, morphology is not the same as anatomy. Okay, so form and shape is not the same thing as what parts are present. You know, the shape of an automobile is not tell you what parts are under the hood.
Yeah, so almost all vertebrates have exactly the same parts. They're just shaped differently.
Now, that's not so. Fish are vertebrates and they don't have lungs, with the exception of lungfish.
Actually, most lungs have some kind of gas.
So that's not true. Yeah, but that's not a lung.
Do you know how a swim bladder works?
A gill is not a lung.
I didn't say anything about a gill. Do you know how a swim bladder works?
Yes, and there's no reason to believe a swim bladder evolved into a lung.
Actually, there's strong reasons to believe that a swim bladder evolved out of a lung.
Do
you know how a swim bladder works?
I know all that, but the point I made was
explain for the audience how a swim bladder works.
Elongating a thigh bone
Please explain for the audience. Please explain for the audience
to the anatomy of the organism.
Please explain for the audience the way.
And so these kinds of changes
please explain for the audience
support for evolution theory, because they don't change the anatomy of the audience. Please stop talking over me, sir.
You're the one talking over me. Please
I've asked you a question for 30 minutes.
Explain for the audience. Explain for the audience the way a swim bladder works.
I want to explain the reason for my question.
You've already said it
Was that-
15 times.
No,
I've already
I
answered it 15
no, I didn't.
times.
No, I
You
didn't
brought up the
actually
lung.
answer.
You brought up
Please.
the swim bladder. Please
Okay,
explain
so
to the audience the way a
the
swim
point of
bladder
it-
works. You
the
don't
point-
know
I
how a
don't need
swim
to
bladder works,
explain
do
that.
you?
I don't
Just
need to
admit that
explain
you don't
that.
know how it works. It's fine.
I do.
I'll
It doesn't matter. That doesn't
I'll
make
tell
it.
you how it works.
Look,
You'll be
no,
very
listen.
impressed.
Okay,
You'll be very impressed by the fact
if
that it
we don't
works
get control of this
exactly
room,
the same as
you
a
can't talk
lung.
over somebody constantly, man.
Apparently
I
I
just want to make
can.
a point.
Apparently, I can talk over
So
you
why don't you
because
come back
you
when you
won't
know how to
answer
debate?
any questions.
You come back when you know how to debate.
I'm not debating. I'm discussing.
No, you're not. You're talking
discussing
over somebody
poorly
constantly.
because
You're
you
under
won't
fire
stop
because of
talking.
a
You
question.
won't let me answer questions without
No,
interrupting.
I asked you
You
a
won't
question.
answer questions without interrupting.
You take
You
20
don't
minutes going
talk.
around it without answering the question.
I answered it
Evolutionists
in
love to do this.
detail,
Okay?
specifically.
The point
By
I'm
the
making,
way, Standing,
when
I told you
ask
this was going to
question-
happen.
Right.
Okay.
I understand your kind. I've been through this with you guys a million times. Ask a question, takes 20 minutes to get an answer. You'll go around and around and around and around, but you won't directly answer a question. That's because evolution theory is garbage. And I'm trying to get to that, and you won't let me do it.
Okay, why don't we get one last thought from Crispr on that?
So
No, I
let me-
just want to hear him
I
explain
want to
how the swim
provide,
bladder works.
before his denial of what evolution posits, I put forth this argument about single nucleotide polymorphs. Okay, so this myth of evolutionists that single nucleotide polymorphs, the majority of them aren't destructive, is a myth. And here's a science paper. I'll post the link to you in the Hangout. Okay, which discusses the effects upon random genetic mutation by single nucleotide polymorphs. And the effect of it is that the majority of the time, when it affects a codon, it's going to replace an amino acid with a different amino acid. Now, because there's only a very limited number of amino acids which can be substituted with a different one, and the protein not change in its folding, that's limited, very limited, in fact, rare, because the vast majority of substitutions that change an amino acid from one type one to another will change the amino acid, and that's going to affect the folding of the protein. It's going to have a negative effect on the protein's efficiency, or it's going to make the protein completely functionless, and little machines in the cell chew it up and recycle the parts. So that's this evolutionist myth that the vast majority of mutations are not deleterious. That's an absolute myth. And protein codes prove this, because the vast majority of substitutions which occur to protein
Your question's clear at this point. Can I answer it?
provide a negative effect to the protein, and that is the cause of virtually every disease in the human genome.
Okay, and then and CRISPR, it's his it's his contention that the majority of mutations aren't deleterious, or like what would be your response to what Neff said here?
He's provided a source that's referring to mutations that occur within coding regions. Coding regions represent a very small percentage of the genome, and the majority of mutations that affect phenotype are affecting regulatory regions, not coding regions. So
If
I
you
agree.
mess with regulation, what are you messing with there? So it's going to be
okay. So you agree then that your entire point is irrelevant because the majority of mutations do not occur to coding regions.
No, not at all.
Hold on, let me finish my thought. You get 50 minutes to finish yours, so I'm gonna let me finish mine. You provided a source that talks exclusively about changes to coding regions, and you said that on that basis, because the majority of changes in coding regions cause problems for proteins, that therefore the majority of mutations everywhere in the genome must also be bad. That obviously does not follow.
Absolutely follows. To say it doesn't follow is
how much? What percentage
nonsense.
of the genome? What percentage of the genome is coding?
About 3% or less.
Okay. So you provided a source.
I just want to clarify one thing. So, CRISPR, are you saying that non-coding DNA absorbs these mutations and makes them absolutely neutral, which means there's no deleterious effect on them if it's a mutation in that region? Is that what you're saying?
I'm not even there. I'm not even there yet. What I'm saying is that providing a source that says the majority of mutations to 3% of the genome are deleterious, that does not in any way suggest what what the percentage of deleterious mutations in the remaining 97% of the genome might be, right?
Well,
You
I think it
can't
comes
get the
down
one from the
to
other.
I think it comes down to whether or not, you know, these non-coding regions do play important roles. I think that would
Certainly it
bring
does.
us into the
Certainly
junk
it
DNA
does.
argument,
But
because
can we
we've
all
hashed
agree?
this so many. I think that's where we'd be heading then to
Right. No, sure.
determine
But
whether or
can we
not the
all
vast majority of deleterious. Go ahead.
can we all agree though? Can we all agree though that saying that most mutations to 3% of the genome are deleterious doesn't say anything about the deleterious nature or not of mutations in the other 97%?
Absolutely not. No, it follows logically that if it damages protein codes, if it occurs to a regulatory sequence that regulates the expression of proteins over time, especially, or has spatial relationships with each other over different loci in the DNA, it's absolutely, I think it's a no-brainer that it's causing deleterious effect there too. It
Okay.
doesn't make any sense.
Can I jump in? Can I jump in and explain why that's not true?
It doesn't make any sense whatsoever to accept that substitutions in protein codes cause a deleterious effect. But when you change a bit of data in a regulatory sequence or any other sequence, it for some reason, we have no reason to believe that it doesn't cause a deleterious effect there. That just makes no sense. That's me.
Please don't repeat yourself again.
Okay, guys. Okay, so
to me, that's like saying I can shoot a car in the front of the vehicle on the front fender, and we see that it causes damage there, but it doesn't logically follow that if I shoot the car in the rear that it's going to cause damage there. It just makes sense.
Awesome. And then thanks, Steph. Okay, and then to Crispr, if you want to explain why you believe that, say, a mutation in say a regulatory area of the genome in the non-coding region, why that wouldn't be deleterious. Go ahead.
Well, it's fortunate that Neff already gave the reasons why protein coding mutations are deleterious and exactly how those are not relevant to regulatory regions, because the problem with changes in the protein region is that it messes with the folding of the protein. But changes in the regulatory region don't change folding of the protein. They only alter expression levels and timing of expression and etc.
But that's not deleterious if that information is damaged?
That can be deleterious or neutral or beneficial, depending on the nature of the change and the nature of the environment that the creature is in.
Example of it being beneficial, please?
The change in the length of the femur.
No, expression of the proteins.
Yes,
how much protein is made, or
that specific mutation works by changing the timing, not the timing, the expression level of a particular protein. I don't remember which one off the top of my head. I'd have to look it up. But it is a regulatory change that alters the length of the femur, which is exactly the thing that we're talking about here. It is an example going exactly to the question you've asked.
I don't see how that logically follows. On top of that, it's been discovered that the genome is so complex, in fact, that there are codes written on top of codes. In fact, some of the base pairs that make up the sequences that code for proteins also constitutes codes that regulate how those proteins are expressed, and because of overlapping and nested genetic information. So when you, if you're changing out a codon in a protein coding sequence, you're likely also almost certainly changing bits of data in a regulation sequence that regulates the expression of the code. So you're causing two types of damage. It's compound, in fact, because the very base pairs of the codes for the proteins,
you've already stated this.
Also, they're shared by different sequences that regulate. So that's
your point is clear. I can respond. Okay, you don't have to repeat it 3 times. I can just respond immediately. Okay, so I've already stated that changes in coding regions are very likely to be deleterious. So saying that in addition to serving a coding role, that some nucleotides also provide a regulatory role, and so changing them is going to be bad, would seem to be covered by the fact that I said that changes to coding regions going to be bad. However, the multifunctionality applies to a very small percentage of the genome, specifically the functional portions.
What scientific reason do we have for believing that the majority of mutations to non-protein coding genes is not deleterious?
The fact that we have the genomic evidence to support it.
Such as? What is this evidence?
Well, you can look at the 1,000 Genomes Project and you can see the levels of variation between people, including parent-child pairs.
Well, we all know that human beings are not all clones of each other, so I don't see how this is an answer. I'm just wondering what scientific reason you have for believing that mutations to non-protein coding genes, that is the vast majority of the DNA, which is now known to code for RNAs, by the way.
Not all of it.
87% is the current science on it.
Oh, yeah, that's not most of that spurious transcription.
87% is believed to code for proteins, and that may be increased with continued research.
No, not code for proteins.
I didn't say it codes for proteins.
You literally just did. You may have.
No, I said no. I said, well, maybe I did. I said originally said that 87% of it is now believed to code for RNAs, which are regulatory snippets of regulatory information, and other things too. So this is information obviously being transcribed by the cell. It follows logically that it's used by the cell for something.
That
we just actually don't know what it does.
Logically.
So absolutely it follows, just like words in a book that you can't read because they're in a different language doesn't mean they don't have a purpose. So since it's information that's being transcribed by the cell, what scientific reason do you have for believing that the vast majority of proteins mutations that take place to the non-protein coding areas of the DNA, which are now known to code for RNAs, which are
sure? If I could be allowed to finish the thought that I was already in the middle of when you started talking,
are not deleterious. What reason? What science is behind that?
So if I can now finish the thought that I was in the middle of when you interrupted.
Well, I'd like you to answer that question.
Yes, this is the exact. No, that was actually the question I was already in the process of answering when you interrupted. So if you could not interrupt more so I can actually answer it instead of commenting on your interruption, that'd be great.
Okay, go ahead, Crispr.
Look at the 1,000 Genomes Project. What you see is you can actually see mutations between parents and children, and you can see where those mutations are the most common, where they exist, and what regions they affect. And you see that you have a lot of mutations in all of these non-coding regions, including all of the regions that you're talking about, and there are no significant phenotypic differences. There are no phenotypic differences that are attributable to these mutations. You can also look at the
both humans so
well. So if you admit that there's no phenotypic change, you admit that the thing you had just finished saying is wrong.
No. All you're saying is they're both fully human. That's all. But that's that doesn't answer the question. We know they're both human. Okay, so but you haven't given me a scientific reason for believing that a mutation, the majority of mutations which happen to the non-protein, you admit that the majority of proteins, that mutations that occur to protein coding genes are deleterious. But then when it comes to the vast majority of the DNA, which is 97% or possibly 98% of it is codes for RNAs and other things, so it's doing other things, regulatory function and whatnot. You haven't given us a scientific reason to believe that when mutations occur to that information, that's not deleterious. What scientific reason do we have for not believing that it's deleterious there as well?
Sure. The scientific reason we have for believing that it's not deleterious is that we have strong evidence that the upper limit of functionality of the genome is about 20%, and it's probably only about 12%.
Okay, so that's just saying we only know what about 12 to 20%.
No, you've misunderstood me. You've misunderstood me. We don't know what all of the functionality is that exists within that 12 to 20%. But we know that only 12 to 20% can be functional. Whatever the functional classes might be, they must exist within this 12 to 20%.
Look, this is
This
what's
is
wrong with what you
this
just
is
said.
demonstrated.
87% of the DNA that doesn't code for proteins is known to code for RNAs. RNAs are critical pieces of information used by the cell for regulatory purposes and other things.
You want me to
Okay,
explain the
When you
biochemistry of that?
are you telling me that you don't believe that when a mutation occurs to a sequence that codes for an RNA, it will have a negative effects? No negative effects there. Right?
I'm saying that I know through demonstrable science that the overwhelming majority of transcription is spurious.
But back to the point, 87% of the DNA that does not code for proteins is known is to code for RNAs. RNAs play a critical role in the cell.
Yes.
So what is the scientific reason for believing that if you change an RNA because you mutate it, it doesn't have a negative effect? What science would cause us to believe that?
Because we know through demonstrable science the overwhelming majority of the stuff you just said is spurious transcription. Because every nucleotide.
What do you mean by spurious transcription?
Non-functional transcription.
Okay. So you're saying that it's known that most of the RNAs produced by the majority of the cell are garbage in the cell and they have no function?
Yes.
That's not so.
Well, let's be clear about what I'm saying. Most of the identifiable transcripts by sequence.
Now you just misrepresented the heck out of the science.
The cell will transcribe in greater number those transcripts that are functional because there are systems in place to cause the transcription of those functional transcripts. Right?
All you've done is say that you don't believe that they're functional unless they're heavily transcribed, and that's not.
That's actually not what I said.
Anything.
That's actually not what I said.
That's not even scientific. That's not even logical.
That is actually. There is a correlation between function.
What's logical is that because 87% of the DNA is codes for that doesn't code for proteins, produces
How do you
RNA.
demonstrate function?
So RNA, it follows that it is functional, or else the cell wouldn't be producing it. True, we don't know what all these RNAs do.
Okay,
that doesn't give us reason to believe they're nothing and they do nothing. So just because we don't know what they do doesn't imply they have no function. The truth of it is,
you've made your point. You haven't finished my point.
You don't need to repeat it. I haven't finished my point. The worst part of this is that that's like believing that the cell fills itself with garbage all the time and evolution wasn't able to stop it. That doesn't make any sense. If this was actually producing non-functional RNAs and all of that stuff was just being put out into the cell having no function, evolution theory was capable of cutting down the protein codes to only 3%. You know about the junk DNA paradigm. It's dead now, right? You understand it?
So actually junk DNA is alive and well.
No, it's not. Scientists, the most prominent geneticists in this world, are giving it up already.
That's not true.
You're behind the times. So
you're just wrong.
No. So this is where evolutionists put their myth into science, where we accept that the majority of mutations that occur to protein codes, just because we know what the protein does, or we don't even know what all proteins do, by the way. You know that? So we accept that that's deleterious. But when it comes to the 97% of the cell, of the DNA that doesn't produce, that is 87% of that is transcribed into RNAs, which we know are functional.
No, we don't know that they're all functional. That's the point.
Because we don't know the function of that RNA, you say we should assume that the mutations to those codes that produce those RNAs aren't deleterious. And that's a non-scientific, mythological way to approach science. And that's why evolution is myth.
I've got several things to respond to in that. The first thing is that you need to actually demonstrate that each transcript is functional.
No, I don't.
Yes, you do.
No.
The second thing is that if you want to say that something is functional, then it must have an effect on phenotype, because if functional doesn't mean has an effect on phenotype, it doesn't mean anything at all. Now,
not so regulatory. You know, biochemistry.
yes,
RNA
regulatory regions
mhm.
have an effect on phenotype.
Regulatory. Well, we just don't know what all those regulatory RNAs are doing. That's all we don't know. That doesn't
If you
mean
don't know that it's
we are to
doing,
believe
hold
that it
on,
doesn't
hold
cause
on,
damage
hold
to
on. If you
RNAs,
don't know
okay?
that it's doing
It's
anything,
nonsense.
if you don't know what it's doing, you don't know that it's doing anything. You need to demonstrate
No,
that
no,
it
no.
actually does something.
That's anti-science right there. See, that's like saying,
No,
well,
science
because a computer,
says,
it's nonsense. That's saying just because we don't know what those RNAs are doing, they're obviously junk and they don't have a function. That's nonsense. The fact that they're transcribed by the cell implies they're transcribed for a purpose because genetic information is prescriptive.
no. So the thing is that in science, you have to have a null hypothesis that can be falsified. If you set the null hypothesis as function, you can never falsify it. If you set the null hypothesis to non-function, then you can falsify it. Now, that doesn't mean that you assume that there is no possibility of function. It means that you assume that there is currently no evidence of functionality.
Okay, so
However, when you see variance between individuals that eliminate transcription in particular regions without any effect on phenotype whatsoever, the lack of effect on phenotype means that that thing has no function, because function means effect on phenotype, and if there's no effect on phenotype by having it or not, it's non-functional by definition.
Okay, I just want to ask one
not
simple question.
non-functional if you
I
don't.
just want to ask one question to Crispr and then let him respond, and you can say what you're saying. I'm genuinely interested. Like, let's just say most of it was still truly junk, and that this junk DNA paradigm has not been overturned. Why would the cell actually bother with it? Because
that is such a great question.
Well,
I'm
just want to, because I'm just wondering, wouldn't such an activity be just a complete waste of energy and resources, and then natural selection would have eliminated this long ago? Go ahead.
Yeah, so no, that's a really great question. That's absolutely a great question to ask. So the thing that you have to understand about transcription is that it starts on the basis of affinities for particular enzymes or particular motifs, right? Now, these motifs are short and they're common, right? And they're motifs because you don't need an exact match. And the affinity varies based on the particular microenvironment of the cell, the acidity, all this other stuff, right? And so the point is that all of this stuff has a tendency to kind of just happen on a stochastic basis, which, by the way, we can demonstrate this by looking at regions that we know to be random. Now, the problem is that the more difficult evolution makes it for the cell to start transcription of a region, the more costly it is to transcribe everything, including the stuff that you want transcribed. Now, you might say that that's worthwhile, but realize that by degrading all of the spuriously transcribed bits, because you're going to degrade all of the RNA that's made, functional or not, you're going to degrade it anyway, and you're going to get the overwhelming majority of the energy back when you do that, right? So the cost to the cell to have these spurious transcripts is actually very minimal, because they're transcribed at such an extremely low level. You talk about this 87%. That 87, and actually I think that 87%, that sounds higher than what I've read. I remember a slightly lower number. But I'll give you 80%.
87 is in code project.
Yeah, I have various problems with encode. I don't care.
I know every evolutionist does.
I'll take 87%. It doesn't matter. 87 is fine. That's fine. Is that these 80 of this 87%, the overwhelming majority of it had 1 or 2 reads in the entire run of the experiment. That means that one cell had 1 copy. or maybe 2 cells had a copy, or maybe 1 cell had 2 copies. Now, you can say that you can have functionality at low levels, but low levels is usually still measured in terms of reads per 1,000 bases, right? So you would expect, at least a read per, not 1,000 bases, per million bases. You would still expect 1 read, maybe 10 reads per million bases, for a very low level of functional expression. So when you have 1 transcript at all, that's indicative of spurious transcription, right? And that is the majority of the transcripts found in this 87%, if we're going to go with that number. So the simple fact of the matter is that not only is there strong evidence to support the case for spurious transcription, not only is the majority of the energy used to create these spurious transcripts recovered by the cell, not only would doing things to prevent these spurious transcripts cost the cell more ultimately in terms of causing more difficulty in transcribing the stuff it wants to transcribe, right? But there's actually strong evidence in terms of looking at the variance between different individuals to support the notion that there is no function because there's no identified phenotypic difference caused by the presence or lack of these transcripts. So on every single possible testable level, we see that there is no sign of functionality and evidence against functionality, right? Now, if you want to propose a test to identify functionality, right, and do the appropriate science, set your null hypothesis to no function, and then go out and try to identify function, you are welcome to do so. And I wish you luck in doing it. And you might even succeed because I am sure that there are some things that we have not yet found. However, what we have not yet found exists within a quantifiable limit. There is an upper limit on the degree of functionality within the genome, and the upper limit on that is about 20%.
Okay, so you think the upper limit is about 20%. Okay, we'll allow, I think you made your point clear. You answered the question. We'll allow Nephilim to make his point and give his thoughts on what you just said. Go ahead, Neph.
Well, this whole idea predicates on the idea that because we don't know what the function of it is, that somehow it's not going to matter when it's mutated, which I think is a very anti-scientific. It's obviously a very anti-scientific way to approach it. Where is the information that the protein codes are not the information that is used during fetal development to produce an organism? Okay, that information that says this will be a bone, and this will be here, and it will be this long, and it will be exactly this shape, and this will be a fingernail, and this will be a tendon, and it will be located here, and this will be an artery, and it will be located here, and this will be a heart valve, and it will be located here, is a massive amount of information to create a human being during fetal development. Massive. It's not the proteins that do it. It's the information that regulates the cell specialization and the use of proteins. Where is that? Well, that's not in the protein codes. That's in that 97% of the DNA. Okay. Therefore, because the proteins code for a small amount of the information in the whole genome, this vast majority of the genome is information we know it's at least this. It's information that codes for how to build a human body in the womb. Okay, there's going to be a massive volume of information. That's where it is. It's not in the protein codes. It's elsewhere. It's this 87% and possibly other more that codes for RNAs. So if you tinker with that information, it follows logically. You're messing with the information that develops an organism. That's going to have in knowing the effect of random genetic mutations in general, we have only reason to believe that a typical mutation is going to be destructive. Okay, so on top of that, if it's coding for an RNA, we have every reason to believe that it's useful. Evolutionists have put forth this junk DNA paradigm for, they got away with it for 40 years, and they basically held back genetic science for 40 years by putting forth this false idea that the majority of the DNA is junk left over by evolution and it's not useful. Today we know that's not true. It's fetal development information, it's regulatory information, it's also information that regulates what proteins are to be made over what time. The genome is four-dimensional, not just a three-dimensional molecule. The fourth dimension is time. Some proteins in your body are regulated and expressed more while you're sleeping than others while you're during the daytime, and some during phenotypic expression. So to develop an organism in the fetal development while it's developing in your mother's womb means entire sets of proteins and other genes are turned on and off like orchestral instruments in this unfathomably complex patterns that turns genes on and off in groups and individually over a period of 9 months to make an organism in the case of human beings. That's information. And that's not in the protein codes. So this idea that the majority of the, because we don't know what the function of it is, it logically follows that it's not functional and therefore it can absorb all these mutations without having a negative effect is anti-science. That's not a scientific approach. And that's the very thinking of this junk DNA paradigm that held back genetic studies. Here's how it did. Because the evolutionists believed that the majority of the DNA, that is the information that doesn't code for proteins, was junk left over, scientists simply refused to study it for 40 years. We would know vastly more about genetics today if that paradigm had not been held to. So evolutionists, with their anti-scientific belief that just because we don't know what the function of it is means we have scientific reason to believe that it's not doing anything, despite the fact that it's obviously much of it is coding for proteins, RNAs, which are regulatory and other function snippets of critical information in the cell. See, this is anti-science. Evolution theory is anti-science. To believe that it doesn't have function simply because you don't know what it does. If we applied the same thing to protein codes before protein codes were discovered, the evolutionists probably would have said, well, it's junk. So this is the way evolutionists think. It's really it's an attempt by evolutionists to fix the problem with their theory, their myth that mutations not only are not destructive in general, but actually design fantastic things. The heart valves, your brain, your liver, your pancreas, everything, your eyeballs, all of this is designed by genetic information. And so the evolutionists, in order to fix the problem that has been discovered by 90 years of research, that mutations in general cause destructive effects to organisms, evolutionists have put forth this mythological idea that mutations in general are not negative. But science has overturned that with a century of research. Evolutionists simply refuse to accept what science shows us about the effects of mutation because they believe the opposite is true. Not only is mutation not destructive, but it's actually this grand, fantastical designer that builds eyeballs and heart valves. And that's just absolutely anti-science.
Okay, perfect. Okay, Crispr, you can go ahead now.
Yeah, so firstly, I just want to repeat this because he seems to keep missing it. We're not saying that it's not functional because we don't know the function. We're saying it's not functional because we know it's not functional. We can demonstrate non-functionality.
That's not true.
You're just wrong. I'm sorry, but you're wrong.
That's not true.
The thing is that you're not familiar with the literature.
No, that's just not so. Scientists have not studied what every.
Sorry, I am not done. You said a lot of stuff and all of that. I'm going to keep going.
They just don't know that.
So, for next thing is that all DNA codes for RNA by definition if it's transcribed. That doesn't say anything about whether or not that transcription is spurious. The fact that it's transcribed makes it code for RNA. That's just a tautology almost, right? So you're not getting to the argument of whether or not it's spurious by just saying that it codes for RNA. Just say that it's transcribed. Save yourself the breath. Next thing, developmental regulation exists within the regulatory space of the genome, which at best is about 15% of the genome. It's probably a lot less than that. Again, we know this because we know how regulatory regions work. We don't know all of the black box details, but we know the approximate sketch, so we can identify them.
That's not true either.
You're just wrong. I'm sorry,
No, I'm
but you're
sorry.
wrong.
They just don't know what most of the DNA does. They don't know.
No, they know it does
The
nothing.
claim that it does nothing, that we know that it does nothing, is not true.
You're mistaken.
No, I'm not. Sorry, that's not science.
You're not familiar with the literature.
No literature says that.
There is literature describing how it is that we know what noise looks like.
The literature does not say that it is
transcriptional
known not to have
noise-
function.
If we know it's noise, then we know it's not functional.
No, the research doesn't say it doesn't have function.
No,
It says we
that it's
don't know.
noise.
It
No,
says we don't know what it
it
does.
says that it is quantifiably noise.
No,
And noise is definitionally non-functional.
no. If
Then you
papers
are
don't say
wrong.
that.
You're admitting that you don't know the literature and that you're mistaken.
No, I'm sorry. You can't provide that literature either. The science papers do not say the majority of the DNA is junk. That idea has been given up. The junk mutation DNA idea is dead. Scientists are acknowledging in droves today that idea is false. Let me give you a couple of statements to that fact, okay? The ENCODE project, Professor Young says, almost every nucleotide is associated with a function of some sort or another, and we do not know where they are and what binds them and what their associations are and more. This from Tim Garris, one of the senior researchers. They say this also, let me see here. Ewan Burney, 39, eminent European geneticist, says the junk DNA paradigm is dead. He says this: should we be retiring the phrase junk DNA now? Yes. I think this phrase really does need to be expunged from the lexicon. It was a slightly throwaway phrase to describe a more interesting phenomenon that were discovered in the 1970s. I am now convinced that it's not useful in a way of describing what's going on.
To respond to, and I
haven't and hold on, I'm not quite done. J. Maddock, cited by W. Gibbs, and The Unseen Genome, Gems Among the Junk, Scientific American, 2003, says, the failure to recognize the full implications of this, particularly the possibility of the intervening non-coding sequences may be transcribed parallel information, may well go down as one of the biggest mistakes in the history of molecular biology. Linda Walgrup, molecular biologist referring to junk DNA and viruses, retroviruses, says, the molecular taxonomists who have been drawing up evolutionary histories of phylogenies for nearly every kind of life are going to have to undo all their years of junk DNA-based historical constructions and wait for the full implications to emerge before they try again. I could provide others. Okay, so the junk DNA paradigm is dead. You can continue to hold to it all you like, but if the game is over for junk DNA.
Junk DNA is still the predominant view of the genome.
It is not.
You're quoting from a select group of scientists who are in a clear minority. Now, yes, you can keep stating things that are false, but I'm going to keep talking anyway. So when you reference the ENCODE project, when they talk about something being functional, what they mean is that it's involved with anything that anywhere else looks like something that might be functional, and include in that anything that's even anywhere nearby. They have like a 2 kilobase window of any biochemical recognition. That is not evidence of functionality. That is at best evidence of the possibility of functionality.
Transcription is evidence of function.
No, it's not. To say that it's evidence of functionality means that you don't know what transcription is and you don't know what biological function is.
That's ridiculous to say I don't know what transcription is, because you really want to believe that? So It's anti-scientific to say if
Do
it's
you know what spurious
transcribed,
transcription is?
if it's transcribed by the
Do you know what
cell,
spurious
that
transcription is?
this implies that it has
Do
no
you know what
function.
spurious transcription is?
It's obvious that if it's transcribed by a
Do you
cell,
know what spurious transcription
that
is?
it's done so because in all likelihood it
Do you know
has
what
function.
spurious transcription is?
So that's
So
completely
you don't know what
anti-scientific.
spurious transcription is.
It's an anti-scientific
So you
approach.
don't know what spurious transcription is.
It doesn't have to be transcribed
Since
constantly.
you don't know what spurious transcription is,
It doesn't
you don't
matter.
understand the
That
process of
doesn't help.
transcription. And if you don't understand spurious transcription or the process of transcription in general, you don't understand how
Look,
this stuff works.
don't tell me what I understand and don't understand. Obviously, I
Explain
know what
spurious
transcription
transcription.
is. So
Explain what spurious transcription is.
it's an anti-scientific way to approach it, to say that because we know it's transcribed, for some reason to continue to believe that it has no function. If the cell makes a copy of the information and puts it off into the cell, it likely does so because it has function. We just don't have a scientific reason to believe it doesn't. That's a completely anti-science way to approach it.
So you can't explain what spurious transcription is.
I've already addressed it.
What's spurious transcription?
So what is a formalgamation of a protein code? Can you tell me? See, we can ask each other questions like this. That doesn't prove that you're right about anything.
Relevant to the point.
You're asking these questions. Evolutionists do this all the time. They think this gets out from the logic, the reasoning behind scientific thinking, and it just doesn't. It doesn't fix anything.
Let me explain to you what spurious transcription is and see if you can't cotton on to the relevance. Spurious transcription is what happens when sequences at random under stochastic processes within the cell are transcribed. That means transcribed at random. That is spurious transcription. It is a known phenomenon that can be demonstrated to occur in all cells at all times. The fact that spurious transcription occurs and occurs at the levels that it does implies that most things should be transcribed at extremely low levels, which is in fact what ENCODE found. They found that most things are transcribed at extremely low levels, which is consistent precisely with spurious transcription and not consistent at all with functional transcription, because functional transcription occurs at higher levels than spurious transcription in all demonstrable cases.
Do you understand?
There has never been a case of any functional RNA having a function at a level of transcription as low as the majority of the transcripts found by ENCODE. Now,
do you understand this? Let me ask you a question. Do you understand that if the information that that develops an organism during fetal development is not the protein codes, and in that being so, then it's part of this 97%, and it's not going to be transcribed heavily after the organism has been fetally developed. Its job is done. Do you understand? So that doesn't mean mutating it is going to have no effect. What it will have an effect is it can cause birth defects, deformities, and
I'm not sure you realize this or not, but what you just said is that if you removed all the protein coding sequences from an embryo, that it would still develop just fine. Because you said that the embryo doesn't need protein coding sequences to develop.
Don't put words in my mouth. That's nonsense. I didn't say any such thing. I said the information that regulates the cell division, cell specialization, and the use of proteins to build structural designs that make an organism during fetal development is not in the protein codes. It's in this 97%.
But now let me ask you a question. Let me ask you a very simple question.
You're willing to believe that mutations to that information is not going to be a negative, not going to be a negative thing. And that's an unscientific way to approach science. And that's just like this junk DNA idea.
Stop talking for a few minutes.
That is held back
you
science.
Can listen to this question and you can understand. Oh yes, let's talk about that lie that you said. Let's talk about that lie that you said.
You don't have to talk over
me. Lied twice now.
You don't have to talk over me constantly.
Yeah, I do, because you're lying. You're lying right now.
No, you can let me finish my sentence, okay?
I let you finish your lie?
Like this junk DNA.
Lie?
Look, we have to be adult about this, okay? If you can't handle acting like an adult.
And I'll let you talk.
It's just like this junk DNA paradigm all over again, where we have no reason to believe that it's functional just because we don't know what its function is, even though it's transcribed by the cell. So mutating it's not likely to
We
cause
have direct
any harm.
reason to think that it's non-functional, so we think it's non-functional. Thanks.
No, we don't, because you don't know what parts of it fetal development information and what's not.
Actually, yes, we do.
No, they don't.
This is the fun fact. Is actually yes, we do.
No, they
we can
don't.
actually identify.
No, that can't be observed. That
Yes,
process can't be observed. No,
because we can monitor transcription at a cellular level during development.
false.
You're not familiar with the current
No, we
literature.
don't even have a camera that can watch it, sport.
Are you kidding me?
The technology isn't there.
We've had it for 10 years now.
No, you can't observe what's going on in the phenotype during fetal development and measure what's happening with every regulatory sequence that's in what it's doing. Nobody's able to do that. We don't have that technology yet. What the hell? What kind of pseudoscience Martian program are you talking about? They don't know how to do that. That's beyond man's technology. We won't have that possibly for decades, maybe decades upon decades. I don't know what the hell you're talking about.
Yeah, so actually we've been able to do that for a while now.
No, we can't do
I'm just not familiar with the current literature.
No.
See, this is the hilarious thing, is that you're so out of date.
No.
You're talking about
So,
science.
We can see that certain proteins are regulated more frequently during certain stages of fetal development. That's as far as it goes. We can't say what information is used and what's not all throughout this process of fetal development. They don't have that technology. They don't even know what 97% of it's doing. Okay, so they sure don't know.
Most of it's not doing anything.
They sure don't know. Well, that's your junk DNA paradigm that's been overthrown.
No, that's the evidence of modern science.
So your statement that they can monitor and know what 97% of something you say they don't know what it's doing is doing, you just contradicted yourself. What flavor is that foot of yours?
I'm sorry, I was finding my link about them monitoring development and
if I can just ask something or add something, wouldn't, in regards to what he's saying about the embryonic development, wouldn't experimentally testing, say a DNA sequence in, each of these unique windows Neff is talking about, in which the sections of DNA are actually used and then say silenced, wouldn't that be an enormous experiment? Like, wouldn't the types of experiments done to actually prove for certain all the many functions that are going on in those various stages on a molecular level in the embryo?
So the way that you do this is that you draw correlations between genetic variants on the basis of developmental abnormalities. Then you label those genes so that you can monitor their expression in vivo during development, which is exactly what people do, and that's how they've been able to figure a lot of this stuff out. Also, you can do single cell transcriptome analysis, including in real time. You can do various other things that Neff is just not familiar with because he hasn't been following the current science.
It's not true to say that they know what the function of it is and know that it doesn't have function. Either one. All we know is that the majority of it's transcribed. So you've completely misrepresented the science. But here's a new article. Here's a relatively new article. 2018, April 11. Scientists discover a role for junk DNA. Researchers at the University of Michigan Life Science Institute. This is at Science Daily, a very pro-evolution, secular science site, which cites their information from papers and interviews scientists. So this is nothing coming out of the top of their head. They only report what's going into the journals. They say this: University of Michigan Life Sciences Institute and Howard Hughes Medical Institute have determined how satellite DNA, once considered to be junk, plays a crucial role in holding the genome together. Their findings, published recently in the Journal of eLife, indicate that this genetic quote-unquote junk performs the vital function of ensuring that chromosomes bundle correctly inside the cell's nucleus, which is necessary for cell survival. That's just one of the many. So over and over and over and over, scientists are stating that the junk DNA paradigm was a neat idea, but it's not holding up. because we continue to find more and more function in this majority of the DNA once claimed to be junk. It's no longer a scientific way to view it, to believe that it's junk and has no function, especially if it's transcribed by the cell to make RNAs that are put out into the cell.
Right.
That
So let me
implies
respond to
function.
it.
So
Let me
you're
respond to it. Let me respond to
holding
this
to
thing
a
you've said.
junk DNA paradigm, tooth and nail, and you won't let go till your cold dead fingers are pried off of it because you need evolution to be true.
So I can actually respond directly to the article you've just referenced. The first thing to understand is that satellite DNA in eukaryotes has a minimal level, which is what we need to look at, of 0.1% of the entire genome. Secondly, the article that you're referencing doesn't say that all satellite DNA is involved in the organization. It just says that some of it is.
Right.
Now,
it's just one more example. That's all. One more of the
right. Now,
continuous
let me
discoveries
finish
of
my
function
thought, please.
in the
Let
DNA.
me finish my thought, please. Please stop talking while I am talking. That means that at best, they have shown that a percentage, that a tenth of a percent of a tenth of a percent of the genome has a function that was not previously known. Now, you cannot continue adding this stuff up and expect, because
What?
you think what you're saying is,
Can't add it up.
let me put it this way, right? We know the kinds of function that can come out of certain structures. So we see satellite DNA. Well, what can it do? Well, it can help with the organization of things. How much of it's involved? Tenth of a percent. And it makes up a tenth of a percent of the genome. So that means, well, I guess that means that in the ones where it only makes up a tenth of a percent of the genome, we've added a very small amount to the functionality. In the species where it makes up 36% of the genome, there's no reason to think that any more of it is involved. And in fact, when we test it, we see that there's not any more of it involved, which means that now we have 35% of the genome or more demonstrably irrelevant.
No, that's ridiculous.
Because
I just pasted
you
a link.
don't understand the science.
No, I just
That's right.
pasted. No, you're in denial of science. I just pasted a link into the chat there to to an article. Some DNA dismissed as junk is critical to embryo development. Exactly what I said. Okay, so the majority of the information that, in fact, all of the information that actually produces a human being is not going to be found in the protein codes. The proteins are the building blocks and the machines that get work done. The regulatory information that is the recipe for how to assemble an organism is not found in the protein codes. Okay, that's going to be in your imagined junk DNA, the 97%, the majority of which, 87% now, has been observed to code for RNAs. Okay, so to believe that it has no function, though it's transcribed by the cell, and knowing that the majority of all this information that codes for fetal development to build a whale in the womb, to build a human being or a rhino in the womb is not going to be in this 97%, and to believe that just because evolution needs to hide all the negativity of mutation, to believe that it's not functional is anti-scientific. I'm sorry, it's just anti-science. It's another way of holding back science. So
yes.
Here's another thing about random genetic mutation that kills.
You said one thing. Let me respond to one thing.
I'm bringing up another topic, and that other topic is natural selection is not capable of.
Can't you just be wrong about one thing at a time, though?
Of course, I'm not wrong. Random natural selection is incapable of detecting all the negative mutations that are accruing in our genome. This one researcher,
you have to demonstrate that they're actually negative, which you cannot do.
I just gave you all the scientific reason in the world to believe it. And the junk DNA paradigm is dead.
No,
we know the regulatory information is not in the 97%. It's observed to code for RNAs. Bam. Scientific reason to believe it's functional.
Nope, no, it's not. It's scientific reason to think that you don't understand what transcription is.
That's ridiculous. That's like walking into a factory and seeing that all these different components here, we know these three over here, this machine makes something. We know what the function of it is. We know what those parts do. But here's these big machines over here, and they're spitting out parts. And, I just don't have any reason to believe that those parts have any function.
No, it's like walking into a factory.
Anti-scientific.
let me respond to your analogy.
But back to the point I was bringing up.
Sorry, you said something silly and wrong. I'm going to
Natural
respond to it.
selection doesn't have
I
the
need moderation
ability.
here.
Natural selection doesn't have the ability to see the vast
Sandy, I'm going to
majority
need
of mutations that
moderation
occur.
here.
Okay, just one second. Okay, just one second. So obviously we know that there's a strong disagreement in regards to junk DNA and function of those non-coding regions. Let's allow Crispr to say one last thing in regards. Is that what you want to do? You want to respond to one last?
I want to respond to his analogy, right?
Pardon?
I want to respond to this analogy, right?
Okay, respond to the analogy.
By percent of the contents of the factory, right? What we're seeing is all these big machineries, and we know what they do, and we're seeing some other machines, and they're doing something, and it looks similar to the stuff we know what it does. We assume that they're doing something important, and we also see like a bunch of dust and like metal shavings on the floor and stuff. A tiny percentage of all the stuff in the factory, we assume that that's waste. That's what these transcripts are.
RNA is waste.
The majority of it is, yes.
And you know that because what?
Because we can see the variance between individuals, such that some individuals don't have any of these transcripts at all, and others do. And also, some individuals don't even have the regions that these transcripts come from.
They don't even know what they do, and you're declaring
they don't do anything. This is the thing that you're not understanding. They
don't
know that they don't do anything because if they did something, there would be a demonstrable phenotypic difference between those individuals where they exist and those individuals where it doesn't exist.
It's not so. Fetal development again.
If
development,
Neff, I got to say this for you again. I'm going to say it real slow. If you have two individuals, and in one individual there's no transcript ever at all, and in the other individual there is transcript, but at this minimal level, that means there's no phenotypic difference, including at the fetal level.
For one individual in one particular
If it doesn't do anything for the one individual, it doesn't do anything for any of them, because its absence has no effect.
They don't know what most of them do.
They know that most of them do nothing.
They don't know any such thing. They don't know what they do.
Yes, they do. No. You're not familiar with the literature.
I asked you 40 minutes ago to provide the science papers that say they know what the vast majority of RNAs that are found in the majority of the DNA do. And you haven't done it yet. Can you?
I can't give you a source to show what they do because they don't do anything.
Right. So you can't you can't back up your statement that it's known that these RNAs are functionless.
I can show you papers that show that they don't do anything.
That's what I'm saying, that the majority of them don't do anything. Oh, no. Now, that's a different statement from we don't know what they do. You claiming they don't do anything. That's not what scientists say. They say they don't know what they do. You are misrepresenting science. You're misrepresenting science for your evolution theory. You're lying about what scientists say about genetics to support your evolution theory. You're declaring that the majority of RNAs produced by they're transcribed by the cell are functionless when the scientists don't say any such thing. They say they don't know what they do. They don't declare it all functionless. You're lying about science for your.
When you can demonstrate that transcription is spurious,
we're done.
You have in doing so demonstrated that it is not functional.
When you have to lie about what science says about,
if you don't understand what spurious transcription is, you have no business involving yourself in this conversation. You're just embarrassing yourself.
Evolutionists are anti-science. They hate real science. They hate what discoveries and
the real science says that the majority of transcription is spurious.
They lie about what scientists say. You've been telling us that it's known that they don't have function, and that is a lie. Scientists say we don't know what their function is. They do not say it's been discovered that they have no function. You have lied.
It's been discovered that they are spurious.
You've lied about genetics for your evolution theory. I don't need to do that as a creationist. I don't have to lie about.
Yeah, you just need to lie about, people not studying non-coding regions. which
Okay,
is a
so
lie.
I don't have to lie about science to support creation. But evolutionists do this all the time, grossly misrepresent to the point of lying, in fact, oftentimes, about what science has to say about something in order to defend a
So are you
failed
denying
idea.
the science of
So
spurious
now
transcription?
the point
So
that
are
I
you
was making,
denying
the
the science
second
of
point that I
spurious
was making
transcription?
is
So
natural
you're just
selection doesn't
lying about
have
what
the
scientists say
ability.
about spurious transcription.
Natural
So
selection
you're
doesn't
lying
have
about what
the
scientists say
ability.
about spurious
Okay,
transcription.
okay.
Natural
So I think at
selection
this
doesn't
point,
have the
we
ability.
understand where both sides are on the junk DNA issue. Why don't we switch gears into whatever this specific topic is, as long as it's unrelated to what we were just previously talking about. Go ahead.
Well, I was pointing out that natural selection doesn't have the ability to detect the vast majority of mutations. They're simply slipped on by. And science research has shown that the rate of human mutation is vastly higher than what it was believed to be, and that rate, which has been a problem for evolution theory all the way back to Haldane in the 1930s. But the idea was called Haldane's dilemma. The dilemma is, since mutations are observed to cause unfitness in organisms as a whole, then how do we fix the problem with the organisms, the species remaining fit when natural selection can't see the majority of them? And certain researchers have put forth the idea that 44,000 children you'd have to have just for two of them to survive, and natural selection select only two of 44,000 in order to keep the species alive over time because of the recruiting of random genetic mutations degrading the genome. This research has continued. Jonathan Sanford has made a big stink in the field of genetics because of his support of that theory that's been put forth, that this understanding that natural selection is not able to fix the problem. And Jonathan Sanford has published a book in 2005, the mystery, genetic entropy and the decay and the mystery of the genome, he cites other scientists in his paper. It's not just ideas off the top of his head. So what's been discovered is that genetic mutation is in general negative. This flies in the face. It's the opposite of what the evolutionist has been saying for 90 years, that mutations are the grand designers of things. We just don't have any scientific reason to believe that at all. In fact, this junk DNA paradigm came into being largely because of this. Haldane's problem, we underestimated it. In fact, H.J. Mueller in 1950 and Kimura in 1968, Nachman and Crowell, J.V. Neely in 1986, Ewan Walker and Knightley in 1999, and others have shown that the sets of parents would have to be as many as possibly 2 million offspring per parents just to get 3.27 organisms that are fit to survive.
We've already actually talked about this.
So this kills evolution theory because mutation is not the designer of anything. It's a destroyer.
Okay, okay, awesome. Thanks there, Neff. Okay, Crispr, go ahead.
So the models used by all of the people you just referenced ignore beneficial mutation entirely because the inclusion of beneficial mutation in those models causes evolution to occur at a much higher rate than that is observed, which means basically that you're wrong. And also, Sanford lied about what Kimura said because Sanford said that Kimura didn't include beneficial mutation because he said that there weren't any, which is a direct misrepresentation, an intentional misrepresentation of Kimura's work. So you're wrong, and Sanford's lying.
No, he didn't say that. He said Kimura says beneficial mutations are so rare you can't hardly measure them in population genetics. That's what Sanford said. That's what Kimura said. So you're wrong about him lying about what Kimura said. He did not misrepresent what he said. So these beneficial mutations, the thing is, nobody knows where they are. Nobody has seen them. I mean, we point to there is the rare exception of, excuse me, a bacteria gaining an immunity. But let's consider, is that actually a benefit? Okay, so a gene gets, hold on, please. A gene gets damaged that causes, as happenstance, happens to cause the ability of the bacteria to survive the influence of.
I'd like to address a lie you just said.
So the thing about that is,
Sandy, I need moderation.
Is that beneficial? Is it genuinely beneficial? Well, the answer is no, because when the organism is removed, taken out of the petri dish and put back in its normal environment, not one where it's cared for and given a specific diet that's not its normal diet, guess what happens? It dies, because it gained an immunity to something in a specialized environment where it's cared for under special conditions with special food, special temperature, special light in a petri dish. But put it back in its normal environment, the bacteria that gained the immunity because of the random mutation claimed to be beneficial.
I want to stay on this thing that you said about Kimura, because it's not true.
It became unfit, and that's not
finish your thought there, Neff, and then
the
allow
other
Crispr to go ahead.
Well, I was going to point out the other beneficial mutation, which causes sickle cell anemia, but that's not beneficial either if you get a disease from it. This is what evolutionists have to offer in beneficial mutation.
I agree with that point. And now, Crispr, you wanted to address something on
so
more.
Kimura said in his actual work, in this formulation, we disregard beneficial mutations and restrict our considerations only to deleterious and neutral mutations. Admittedly, this is an oversimplification, but as I shall show later, a model assuming that beneficial mutations also arise at a constant rate independent of environmental changes leads to unrealistic results. Specifically, those unrealistic results are that evolution occurred at an enormously high rate. So in other words, including beneficial mutations at any level in the model causes evolution to occur at an unrealistically fast level, which you could say means that the entire model must be wrong. But if you want to throw out the entire model, that means you also have to throw out the deleterious side. The simpler explanation is to do exactly what Kimura did, which is the thing that Sanford wants to take advantage of, which is to introduce an element of very slightly deleterious effects to the otherwise neutral mutations, which then has the effect of balancing out the beneficial ones. That is what Kimura's work actually did. If you'd actually read it, which I know you haven't, and I don't think Sanford has either. I suspect, because if Sanford has read it, then he's lying about it. But that wouldn't actually surprise me.
So here's how you've misrepresented science again. Kimura is the one who put forth the idea that because natural selection can't fix the problem, parents would have to have 44,000 offspring and only 2 survive for natural selection.
I just read to you what Kimura said. But you're lying about him.
Wait a minute. No, I just related exactly what he wrote. But the problem is that Kimura put forth this idea because he himself believed that the majority of mutations are destructive. Okay. Otherwise, he wouldn't have postulated a mathematical equation for solving the issue with natural selection. So you've misrepresented Kimura as implying that he didn't believe the vast majority of mutations are deleterious, when in fact that's what he did, and he did, and other researchers that I mentioned, and many others, because that's why they put forth these population genetics models in order to figure out, in order to figure out how natural selection can solve the problem.
You know, I'll interrupt you less if you lie with less frequency.
Look, just grow up a little bit. A little.
Stop lying so much. It's really easy.
We've proven
I can tell you the truth.
Already. Okay, so let me finish my statement.
What Kimura said.
My statement.
I read to you exactly what Kimura said.
Excuse me. The reason Kimura and other geneticists have put forth these population genetics models to try to fix this problem with natural selection is because they accept that the vast majority of mutations are deleterious in nature. Okay, that's a fact. So if you want to believe they're not, you need to show us these beneficial mutations. Where are they? So
let me answer this. The inclusion of any level of beneficial mutations causes evolution to proceed at an unrealistically rapid rate. In order to account for this unrealistically rapid rate caused by beneficial mutations, which people knew occur at some level, because they must occur at least some level for reasons that I can demonstrate mathematically, that means there had to be some offsetting feature, which is what led to the nearly neutral theory. In other words, by going from the assumption that the overwhelming majority of mutations are neutral to saying that the overwhelming majority of mutations are nearly neutral, this introduces a sufficient negative effect to counter the necessary positive effect of the necessary beneficial mutations. Now, if you want to claim that there are no beneficial mutations, that means that there are no detrimental mutations, because detrimental mutations imply beneficial mutations necessarily.
Nonsense.
Then you don't know how mutations work. Congratulations on proving that too.
Nonsense. So where are these beneficial mutations that you believe in this evolution?
At an absolute minimum,
Where are they?
there must be a beneficial mutation for every detrimental mutation, because every detrimental mutation can be reversed, and that reversal must be a beneficial mutation. And even that, even only that, offsets the effects of the detrimental mutations by such an amount that the rate of evolution exceeds all known observations.
Okay, so what you put forth is a belief. It's actually a myth.
No, I put forward a mathematical minimum.
You spoke. So what you believe is that, firstly, a couple of things. You believe that the genome can sustain such an incredible mutation load that as what you're positing, which is quite absurd.
That's
Secondly,
what Burr is positing.
you're positing that it's logical that wherever a beneficial, a negative mutation happens, it's likely that a beneficial one is going to happen to the same nucleotide at some point in the future.
I didn't say it was likely. I said it was possible.
But yeah, that's very unlikely. In fact, that's
yes, and even at that low level of probability, it's sufficient to offset everything else.
Let me finish now. Hold on. And so thirdly, you haven't shown us what these supposed beneficial mutations are.
Changing for moral length.
And they would have to have an effect on, they would have to be building structural designs in the anatomy of living things in order for evolution to be true. So can you show us give us the examples of these? I mean, we know all about the countless science papers, 1 million of them in the journals, 1 million, literally. I'm not making it up. I'm not saying 1 million science papers in the journals that describe negative effects of mutation. Where are these science papers that describe the observed benefit of mutations which affect the structural design of an organism, so we can believe
changing for moral length
build features and cause evolutionary change?
I have to ask this because I'm starting to think that maybe you don't. Do you admit that mutations can cause?
Asking a question is not answering one.
I already answered it twice. Changing for moral length. Changing for moral length. That's an answer to your question. Now I'm trying to ask you a question to figure out why you don't understand why that is an answer.
You have one, okay? And changing the length of a femur, is that somehow? How does that explain the arrival of new features so that we can believe there's a tree of life?
Because all new features come into existence as modifications of previously existing features, broadly speaking, by changing the size and proportion thereof, and occasionally by changing the levels of expression of proteins within those specific tissues.
Okay, so then the keratin at the tail of the stinger and a scorpion, there's no keratin in the tail of that had to be a new feature. Right, the creature had to get the keratin at the end tip of the tail, where there was no keratin in the tail at all before. Okay, so
that's just
what you've just said is not correct. But I just want to know about these millions of mythological beneficial mutations. What's the scientific evidence that there are so many beneficial mutations? And if you point to getting a longer femur, I'm asking you, how does that explain the existence of a new feature, not an existing one?
Because new features come into existence as modifications of previously existing features, specifically by changing the size and proportion.
And I just explained to you that a longer bone doesn't explain the stinger in the tail of a scorpion or its muscle sac. There was no muscle sac.
Also,
There was no enzyme. There was no
Also,
enzyme. Hold
occasionally-
on now. There was no
Yeah.
enzyme that
Look,
produced the poison.
guess what?
That had
I
to
don't-
evolve.
I don't
The
know.
sac that holds the poison,
You're
that's
you're
new.
filibustering.
That had to arise.
I don't know the fine
okay? It
details.
didn't have to
I
be
don't
there.
know the fine
Okay,
details
so
of scorpion anatomy.
These are all new features. They're not something that already exists in the tail. You can't change the shape of the tail and get these things. They're new features, right? So genetic information codes for things. Okay. What is the scientific reason for believing mutations create the genetic information that code for new things?
Because the new features are modifications of previously existing features.
And I just gave you an example
Yeah,
of
Look,
that are
I
not.
don't know the fine details.
And
of
we could go through
scorpion
the
anatomy.
anatomies of a million creatures, and I could do that over and over and over and
Do
over.
you want to go through the anatomy of the
So
tetrapod
okay.
limb?
I just want to know about these beneficial mutations that have been observed.
Because
What makes it
because
scientific to
you
believe?
you accept that mutations can change the size and proportion of pre-existing structures. The accumulation of modifications to the size and proportion of pre-existing structures can build up to evidently new features. This has been demonstrated through mathematical modeling.
Mm-mm. It's been put forth by philosophical constructs. Evolution is a philosophy; it's not science. If you're going to call it
We
science,
also
it
have
needs to be
matching fossil
observable,
records.
testable, and repeatable. So, where are these scientific evidences that mutations add things to body plans that don't exist before? Now,
Well, it
you'll
doesn't-
never. Let me explain the mistake you're making. Evolutionists do this all the time by getting longer legs. If the only change they ever got what was the legs getting longer, they'd simply be a taller human being. It wouldn't change their anatomy not one iota. Okay, so you can't have the tree of life and evolution producing this type of a creature from an antecedent so many millions of times to produce all the various body plans and structural designs and the anatomical features that exist in all the creatures of the world by elongating thighs or making legs longer. You've got to have new stuff, new information.
You got to do more. You're right. You have to do more. You have to do
What
more.
scientific
You have to do more.
reason
You
is
have
there
to
to
do more than making
that
you're
mutations can do
filibustering.
this?
Stop.
I've
You have
asked
to
you
do
a
more.
question.
You have to do more than make the femur longer. You also have to do things like, say, change the size and proportion of the ends of the bone, or change the size and proportion of some other bone, or change the size and proportion of some muscle group, or change the size and proportion of some other structure, or whatever else, and occasionally also change the expression levels so that you're expressing something where you hadn't been before, which would actually explain the presence of keratin in the stinger of the scorpion. But let's not go back to that, because that's silly.
So getting an eyeball means there are structural designs to the eye that didn't exist before, and they didn't exist in a pocket in the skin. See, all of these features of the eyeball, you can't get them by changing the shape of skin. The proteins.
Do you want me to walk you through it?
No, many of the proteins in the eye are not even used in skin. Okay, so see, the point I'm making is
the ones that are light sensitive are.
Changing morphology is not a change to anatomy. Okay.
Yeah,
Anatomy
it's
must change if evolution is true.
It's impressive that
what is the scientific reason for believing that, firstly, what are all these beneficial mutations you claim exist in such great numbers? And what is the evidence that genetic mutations design anatomical features?
So it's really impressive that, skin cells have proteins that are already responsive to light. So
you got to have new proteins to get an eyeball. Can't not just the ones that make skin.
Yeah, the proteins that are responsive to light, the ones that let you see, they're already there.
You need new proteins to make an eyeball. You don't have all those proteins in the skin.
Do you have all of those proteins in anywhere in the body?
Not in the skin.
Yeah, that's change in expression then.
Right. So the feature had to come into existence by structural design.
No,
by
change in expression. By change in expression levels.
Not by changing the skin. You got to have
change in expression levels. change in expression levels.
Evolution
By
requires new
change in
stuff.
expression levels.
Hey, listen.
By change in expression levels.
Evolution
Say it with me. By
requires.
change in expression levels.
Are you?
Say it with me now. Change
Look,
in expression levels.
Are you?
Change in
So what
expression
we've
levels.
got here is the evolutionists denying
Change in expression levels.
because they can't provide evidence
No,
for it.
change in expression levels.
You have to have new things come into being in a location in the body plan for evolution to be true.
So we agree that there are
Can't
already
provide a
proteins
scientific
that are
reason.
light in the skin. And
So
that
what's the
all of
scientific
those
reason
that
for
are needed
believing this?
exist somewhere else and can be brought into the skin by change in expression levels.
So what's the evidence that mutation reorganizes new tissues in a different location in a particular place in the body? What's the
mutation
evidence
doesn't organize it, selection does.
what's the evidence that genetic information codes for new information, mutations code for new information that express features, structural designs in a body plan that didn't exist before? What's the reason for believing that?
When you say that
Scientific.
didn't exist before,
Not philosophical now.
what matters is the difference between one generation and the next. And between one generation and the next, you can never point to a feature and say that it is new, even though you might point to 1,000 generations removed and say that it's new.
Where are these generations or observable examples?
Well, we can observe mutations causing changes in relative expression levels, changes in size and proportionality of pre-existing structures.
Size and shape is not a new thing.
Yes, because between one generation and the other, right, there are no new structures.
Right. Here's the myth. This is where the imagination comes into play. So what is the scientific reason for believing? If it's not just a philosophical idea and it's actually science, you should be able to provide scientific reasons for believing it. What is it?
I can provide scientific reasons for thinking that mutations cause changes in size and proportionality and also relative expression levels of protein, which is exactly the sort of thing required for a cumulative adaptive selective pressure to produce these varying structures over successive generations.
I agree that you believe that. But so what's the scientific evidence that happens?
Well, there's homology.
It's a neat idea, but what's the scientific evidence that happens?
So, okay, well, firstly, you're saying it's possible.
No, I said new things come into being and genetic information is prescriptive. It prescribes that this feature will be designed this way with these tissues organized in this way in this location of a body plan. You've got to also have Hox genes for that too, by the way. So what is the scientific reason for believing this philosophical construct that mutations design the information that codes for features that don't exist in a body plan and will subsequently arise incrementally? What's the scientific reason?
I'm sorry, did you say organizing it?
Can you provide us a scientific reason for believing this?
Yeah, so the scientific reason for believing it is that we see exactly the type of mutations required occur. There is nothing required for evolution that we do not already see occur.
Give us an example, please. These mutations.
Change in femur length.
No. Okay. So
has the alcohol rotted your mind such that you cannot remember something I've said 15 times in this hangout?
You've got to have new proteins to create an eyeball or proteins that are placed in that location. They're not in the skin. Okay.
So changing expression level does that.
Okay, what is the evidence, scientific evidence that how much or little a protein, for example, is expressed, produced or not produced, will do this? Because this is what you seem to be implying.
What's the evidence that a mutation will change the expression level of
protein? No. That changes in protein production will have the power somehow to create how does that create the information that codes for the feature? How does it create the information that codes for where the feature is to be located? That's a Hox gene set. And what is the evidence that genetic mutations actually organize the tissues
Genetic
complexly
mutations don't
over
organize
time.
anything.
So, well, what's the scientific evidence for this?
So the scientific evidence, I'll say this again, is that we know that mutations can change the relative expression levels along with the size and proportion of pre-existing structures. Mutations can also change the relative timing of expression. They can change various other things of that sort, and all of this is demonstrable, and it accounts for every possible required type of change. Every type of change required by evolution is covered by mutation. All you
So,
then need is selection, which we
yeah.
also know occurs.
So mutations put an eyeball where there was no eyeball, right? What's the scientific reason for believing that?
Now, mutations put an increased level of light sensitivity in an area where there was not that same level of light sensitivity.
What's the
Mutations,
scientific evidence for that?
the fact that we've actually seen animals develop eye spots.
The organism already had the genetic information to create light sensitivity in their skin.
Yes,
What's the evidence that genetic mutation developed the ability to create light sensitivity in the skin?
all skin is already light sensitive to some degree.
Okay, so what is the scientific evidence that mutation designed the allele sets that code for light sensitivity in skin?
All skin is already light
There,
sensitive.
there is none, right? Can you acknowledge? You
No,
don't.
I don't understand what your disagreement is. Are you
Well,
saying that skin is not already light
no,
sensitive?
no, you're saying that a lot of things, but I'm asking you for the scientific. I understand you believe all these things, but believing is not. That's not science. You know, I could believe that pink space monkeys are driving Cadillacs around Mars throwing pizzas out the window. That wouldn't make it scientific. That just means I have a
No,
philosophical idea in my mind that I
you asked me how I knew a thing happened that is already true by definition. All skin is necessarily light sensitive.
What is the evidence that mutations do any of the things that you're saying that are capable of placing a new feature in a location in a body plan?
Relative to the prior generation, there is no new feature.
Okay. What's your scientific evidence that mutations incrementally build new features in different locations?
Oh, I'm glad you asked. Change in femoral length.
Okay. So, how is a bone that exists already a new feature?
It's not a new feature.
All right, good.
Relative to the previous generation, nothing is ever a new feature.
Okay, so like the keratin in the stinger of the scorpion tail, there was no keratin before. And like the eyeball, the protein layers that make up the cornea.
Can I just address the scorpion thing?
They're not
Can we
in
do one thing at a time?
They're not in the skin.
Can we do one thing at a time?
I'm just asking for what's the scientific reason for believing that mutations incrementally change anatomy over time? I'll just dumb it down real bad.
So change in expression level would cause keratin to be expressed in the tail of a scorpion.
What's the scientific evidence that that happens? Or has
The fact that we know that changes in expression level will cause the expression of things that weren't previously expressed.
Okay, so it's true that the proteins that produce keratin can be expressed less or more. Okay, so what's the evidence in science that because protein regulation occurs and proteins can be expressed more or less is reason to believe that genetic mutations build the information that causes the expression of keratin in the tail of an organism to build it into a structural design?
It doesn't build it into a structural design. It just causes it to be. Let me finish my sentence, please. It just causes it to be expressed there. The structural design is a product of which cells are expressing it and which times they are expressing it at.
So whenever we observe mutations cause something to be expressed in a body plan where it's not before, that's called a deformity, and natural selection should remove that because it makes the organism unfit. So, but you're positing this is a beneficial thing and it happens incrementally over vast stages of time. I'm asking, what's the scientific reason for believing that?
I'm saying that it can be beneficial or detrimental, and it depends on the precise nature of the organism and the environment in which it lives.
Okay. So the thing is, we need scientific reason to believe that mutations build information, that they build information that regulates information, such as Hox
Mutations
genes, okay?
don't build
and
that.
and
Selection
also,
does.
no, selection only selects what already exists. The change
Yes,
would have to
mutations
occur
cause
by
it to
genetic
exist, and
mutation.
then selection selects it.
Right. So, but the information is built because of mutations taking place. Right.
Built implies multiple steps.
So, what is the scientific evidence that mutation builds genetic information?
Build
things.
implies multiple steps.
Well, that's
build
what you're claiming. Multiple
yes,
steps result
the
in
multiple
eyeball.
steps require selection. Mutation only does one step, which is not building now, is it? Selection builds, mutation creates. It creates one thing, which is one step that selection can then build on.
No, selection doesn't build anything. Selection selects only what exists or not doesn't exist. The
And if it selects new things repeatedly,
the information
that builds something.
No, the mutation would be the builder. The natural selection is the preserver.
Build implies multiple steps.
What is your scientific evidence for believing that genetic mutation builds new structural designs in body plans? There is none.
I can try to answer that, but I need a point of clarification from you. Does the mutation that causes the change in femoral length build a bigger femur?
A bigger is not new.
Does it build a bigger femur or not?
You can't explain the keratin at the tip of the scorpion's tail or the eyeball by getting more skin.
I need your answer to this.
I've explained it already.
You keep using this word build, and I say build is the wrong word to use here.
I've already explained myself. I think it's clear what I'm asking.
Right. So you're apparently...
So what is the evidence for this?
You're apparently either too intoxicated or too stupid to have this conversation, which is what I was afraid would happen. So, I told Standing when I agreed to do this, firstly, that I would only be able to do it for a limited amount of time, but also that we really don't get along.
Yeah, see, I'm not intoxicated. I haven't had a drink of alcohol at all. Not a sip. You're just angry because I'm asking you for evidence for a sign for your
that I've given and you've ignored.
No, you haven't
not understood.
Evidence.
Not understood is much more likely.
You gave me an idea. Getting a longer bone doesn't explain the stinger in the tail of a scorpion or an eyeball or a pancreas. I'm sorry.
Yes, but it explains how bones are different.
Yes, you said you understand it exactly. So you can't get new anatomical features and a tree of life by extending
your brain
bones.
So rotted by age and whiskey that you don't understand how changing bones is relevant to evolution.
So you see, that's the thing. When you point out, when you ask evolutionists for actual
they answer it, and then when you ignore it, you get mad.
Theory that they get very upset and they insult you and everything because they can't provide scientific reason to believe this philosophy of evolution.
Yes, except that I did that, and then you ignored it.
You just said bones can get longer.
I'm sorry. You didn't ignore it. You didn't understand it.
That's insufficient to explain the existence of.
I understand that your brain is not sufficient to understand this stuff, but that's not my fault.
There is no scientific reason for believing. It's simply a philosophy.
Change in femoral length. That is a mutation.
And that doesn't add anything new to the body plan.
Yes, because no individual mutations that would be selected by a natural selection would ever change in any fundamental way the pre-existing body plan from one generation to the next. It is only through successive selection of modifications that you get an accumulation of difference.
Mutations that create longer bones cannot explain the arrival of new features. I'm sorry.
If you have enough of them and you have enough variety of them, they can.
You believe this myth, I understand, but it's not science. It's
what are the differences in the bones between a bat and a human?
Morphology, different morphology.
Yes, so those can all be explained by changes in relative size and proportion. And yet a bat wing is different than a human arm.
And there's still bones, and you can't explain the arrival of the information that codes for new bones.
You just admitted, I'm not sure you realize this, you just admitted
that the arrival of genetic information that codes for new bones, or the Hox genes that will put them in a particular location, or the rest of the feature, you can't explain that by mutation.
You just admitted that bats and humans can both evolve from a common ancestor.
No. They have bone. That
Right.
doesn't provide evidence of evolution.
Yes.
We both have bones.
You
That's
accept
all.
the fact that you can change the size and relative proportion of the bones in the human and the bones in the bat and go back with mutations
won't give
to
you
a common ancestor.
No.
Yeah.
That
That's
won't
what
give
you
you
just said.
in your imagination.
Congratulations.
In your imagination, I
You're
understand.
wrong.
But that won't put the stinger in the
You
tail of a
just-
scorpion or an eyeball in a human being. I'm sorry.
Yeah, human beings didn't evolve eyes. The
Yeah, it won't put
ancestors
it
did.
won't put an eyeball in a mite or any other creature. I'm
I
sorry.
think that most
in a
mites
body plan.
have some kind of basic optical sensor, but that's
I'm
not.
sorry. That's not scientific reason to believe new features arrive in body plans that don't exist before. I'm sorry.
Yes, because new features don't arrive from one
for example,
generation to the next.
It can't explain the thousands of mirrors in the eyeball of a dragonfly that focus light beams to such mathematical military precision.
Yeah, that's change in size, relative proportion, and relative expression.
Those crystalline structures exist in the dragonfly. They don't exist in the creature that dragonflies going down the line eventually supposedly came from. That is a new feature that has to have arrived. And not only that, they all have to be arranged like NASA arranges the glass in a telescope so that it's,
they have to be arranged
precise.
They have to be arranged that precisely.
The precision of that is amazingly precise.
Good God, you ramble on. They don't have to be arranged that precisely to work at all.
They have to be very highly arranged.
But they have to be arranged precisely to work precisely. They don't have to be arranged precisely to work at all.
They have to be arranged to work. They work because they're arranged precisely.
To work at all, they don't have to work. They don't have to be arranged precisely.
They have to be arranged to work. Otherwise, they don't focus the light, silly.
They don't focus the light as well.
Well, in your imagination, there was some defunct precursor. I understand. You believe that evolution stuff.
It wasn't defunct. It still worked. It just didn't work as well.
Well, in your imagination, but you don't have scientific evidence for that, do you? No.
We have evidence.
We
have evidence of what mutations can do. We have evidence of what.
Wait, what's the evidence that mutations can do these things? I've been asking you for an hour.
Control length.
That's not going to explain the arrival of a stinger in a tail of a scorpion or an eyeball in a fish. I'm sorry.
I'm sorry. It's about 2 o'clock where I am now, right? And I've been explaining the same thing to you and having you ignore it or not understand it.
You haven't explained anything.
Out of stupidity for the past 30 minutes.
Okay,
this is why, standing for reference, this obstinance from him, right? And just refusal to discuss things without constantly interrupting me is the reason that I don't like talking to him.
I just don't let evolutionists, put forth all kinds of philosophy and then claim it's science,
No, you just don't understand anything said to you, and so you just ramble off random crap the whole time.
For an hour for the simplest thing about evolution.
Yes, which I gave immediately, and you rejected for no reason.
Get longer. That doesn't tell us how a new feature arises. I'm sorry.
It does. You just don't understand.
Doesn't even code. That's not even where's the information come from that causes the specific arrangements of the components to create the thing. That's new information that would have to arise also. Where'd that come from? What's the evidence mutations build that?
Yes, so this is just like a random gish gallop down all the random different.
No, these are the things that have been asking for an hour. And
you refuse to accept the basics, and then you wonder why I won't explain.
I haven't rejected anything you've said except your belief that mutations do fantastic things.
Yeah.
Nobody disagrees that mutation could make a bone longer or give an organism a deformity. Nobody disagrees with that.
So if you agree that mutations can make a bone longer or otherwise change the relative size, shape, proportion, protein expression, etc., then you accept evolution. Congratulations, I win, you lose. I think this is a good place to call it a night. He's just admitted to all of the pieces and all of the mechanisms and everything required, but he rejects it anyway because he doesn't understand the science. That is the story of the night.
That's the philosophy. It's your philosophy. It's not science.
Yeah, your philosophy is to reject everything that you don't understand.
No, I don't
which is
reject anything
basically everything.
scientific. I told you I agree that a mutation could make a bone longer, can make one
Yes,
thicker
you
too.
you
But
admitted that
doesn't
everything that
explain the arrival.
evolutionists, as you call them, say is true, but you
Evil.
want to pretend it's wrong anyway because
You-
you don't understand science.
You can't get all the features of all the
admitted
living
you're
things
wrong.
of this world
You
by
admitted
changing
you
the
don't get it.
ones the
You
precursor
admitted
had.
your entire life's work is a waste of time and energy and effort, and that you should be embarrassed.
Crispr, let me explain this to you.
In your
You
entire
can't
history.
get, Crispr, you can't
You've been
get all the
doing this for
anatomical
20
features.
years, and you're still just as wrong as when you
Crispr,
started. It's
You
amazing.
can't get all the anatomical features of all the living things by changing the shape of features that existed in something that existed a long time ago. You can't get that. I'm sorry, that doesn't work. It's not even logical.
Except that every time anybody's ever named one that they think is an example of that, people have explained exactly how you can. And
You haven't
they've shown
explained anything.
evidence for it.
You haven't provided evidence. I've been asking you for an hour. All you can tell me is bones can get longer. We know that.
Yeah, because that's the example that I have
That
committed
doesn't
to memory.
explain new features and new locations of body plans.
Yeah, the fact that you don't understand how it explains it doesn't mean it doesn't explain it.
No,
It
you
just
haven't
means that
explained
you don't
anything.
understand the science.
You said
But
you believe it because bones can get longer.
Conveniently, you
Okay.
already admitted to the
Okay,
entirety
Crispr.
of all of the required mechanisms of evolution.
Okay, Crispr, you're welcome to believe that.
Wrong and that I'm right, and that's great.
You're welcome to believe that, Crispr.
I'm glad you now believe in evolution and you
But
reject.
you know what you're up against
creationists.
when you come up
It's
against
great.
me in a debate about evolution. I'm going to ask you questions you can't answer. You know that. At least you learned that.
Yeah, I asked you questions you couldn't answer about the stuff you were actually actively talking about. I still think you don't know what spurious transcription is, but this isn't relevant.
I've already discussed it.
On long enough. Yeah, you discussed how you didn't understand what it was. I know, I remember.
No, I didn't. I spoke about more or less, of a protein being expressed. That's not. I don't know what you're talking about. I addressed it.
I don't.
Before time.
Just for Nestle's benefit, I don't go into random examples of things because I don't find it to be constructive. Because if somebody is so stupid that they don't understand the relevance of change in femoral length, they're never going to admit to anything else. They're just going to reject everything out of hand.
You can't explain the design of all the anatomical features of living things by getting longer or shorter bones, or thicker bones, or skinny bones.
You can explain the entirety of the vertebrate clade.
You need at least 3 types of genetic information for that. You need the information that codes for the features components. You need the information that codes for how they're arranged, and you need the information that tells the organism's anatomy where it's to be located.
Yes, and mutations.
What's the evidence that mutation
changes
does
the
that?
size, the shape, the relative proportion, the relative protein.
For the evidence that genetic mutations build that information. You haven't.
Change in femoral length is an explanation. You just don't understand it.
No, that's just
which is my point. My point is that
that's
you don't understand the science.
That's not new tissues. That's not how they're arranged, and that's not location
there's
either.
Never a new feature or new tissue from one generation to another. It's only through cumulative change.
So you can get an egret from a dust mite by changing what's in the egret, the dust mite.
Probably not too
not a million
examples. You would have to have something that would be a common ancestor to the two of them to get there. And you could get something like an egret,
and you could get a human being from a fish. You can explain the cilia in the eustachian tubes of a woman's uterus with changing what's existing in the fish, right?
Yeah, probably. That's actually not even.
Wow, amazing. Amazing. Wow. What's the scientific reason for believing that?
The fact that we know how mutations work and you don't.
And what's the evidence that mutations do this?
The fact that that's what mutations do.
There's no cilia in any eustachian tubes in the fish.
Are there cilia at all?
There's no eustachian tubes. There's no cilia in eustachian tubes. None of that. See, these features don't exist in the fish.
Those are.
They had to come into
derived characteristics. Guess what they're derived from? They're derived from pre-existing characteristics.
No, there's nothing pre-existing in the fish that's going to make that. You got to have new tissue.
I was thrown by that
in a
too.
specific organization in a
I was
particular location of
confused
the
by,
body
like,
plan.
the eustachian tube in the uterus. What's he talking about?
Uterus, I mean. So, but what's the scientific reason for believing mutations do this marvelous stuff? We just don't have
Because all of the required mechanisms are there.
Really? Explain that. How does mutation do it?
How does mutation cause a
scientific
change in
evidence?
relative size and proportion and relative protein expression?
What are you going to change the size and shape of in the fish to get the eustachian, the female reproductive organs?
You're not going to change anything in the fish. You're going to
Right.
change something in the immediate ancestor.
Right. So it's got to come into being incrementally. What's the evolution? What's the evidence that mutations?
So you have to go from the fish to the thing just after fish, to the
All
thing
right.
just after that, just after
Exactly.
that, after that.
I know.
Now, if you want to go from the immediate ancestor of the first thing with the
Yes. Fallopian tubes. Yeah, fallopian tubes. Right. So I know you believe it takes place in increments in different species over time. I know that. I'm asking what's the evidence in genetics that mutations are capable of doing this?
because we see that mutations can cause changes in relative size, proportion, and protein expression, which is exactly the mechanism required.
So what's the evidence that mutations can do this again?
The fact that we know that mutations cause changes in size and relative proportion and relative protein expression, which is exactly the mechanism required.
And that's evidence that mutations can cause new tissues to arrive by designing new information that codes for new tissues and
mutations
be located in the body plan.
New tissues do not arise in a single generation, nor does evolution expect them
Right.
to.
So what's the scientific reason for believing mutations can do this? So I could ask you, you can't give me a reason because we don't know of any reason for believing. We don't have any reason for believing mutations can do any of this fantastical stuff.
You already admitted mutations can do all of this stuff. You just don't understand the connections.
I think that's really absurd for you to say that. It's kind of childish too, to misrepresent what I say that way.
You admitted that mutations can cause changes in the size and relative proportion and relative protein expression of cells. That's it. That is all you need. That is literally all that you need. The fact that you admit that it happens means that you admit that evolution can work.
I've explained about 60 times already why that's not true. That's not sufficient.
You explained about 60 times
yes.
Your misunderstanding and ignorance of basic science. That is your fault, not mine.
Evolution, it doesn't matter what components, what proteins you start with. Evolution has to create the information that arranges tissues, creates tissues by taking building blocks from the organism and arranging them in a complex manner that results in functionality in a specific location of the gene of the organism's anatomy. There's no scientific reason to believe that. It's a myth. It's a philosophical construct. It exists in your head. There's nothing in the science journals that support that idea. It's a myth.
Yeah, except the fact that mutations cause changes in the size and shape and relative proportion and relative protein expression of cells, which does exactly what is required.
And you haven't told me the reason for believing mutations can do this.
Because we see mutations doing it.
No, we do.
You admitted that we see mutations doing this. I don't know what you're talking about. You already admitted to it. Change in femoral length.
Okay, so look, we can go in circles all night. How does changing the shape of a femur does not arrange complex, numerous types of tissue in a complex manner to provide functionality?
Because that doesn't happen in a single generation.
Right. So what's your scientific reason for believing that the information that does this can be produced by mutation? That's a simple question, sport. Answer it.
Accumulate it through selection.
Okay, so what's the evidence that these mutations accumulate that build the information that does this? So you can't tell us a scientific reason to believe.
Because we see that evidence in selection.
What evidence do we see of genetic mutation being developed that codes for new complex arrangements in body plans? There isn't any scientific reason to believe that.
New body plans from one generation to another. We only see differences through selection over time.
What is the evidence that genetic mutations accrue and build new information that produces functionality? Let me dumb it down even more.
So I just want to make sure here that you are accepting the fact that mutations cause changes in the size and relative proportion and relative protein expression within tissues.
Would a feature that's arising in a body plan have to be functional in some way to be considered beneficial and preserved by natural selection? If it were, it's got to be a benefit to the organism. Isn't that right?
The change in the size and relative proportion and relative protein expression would presumably be beneficial at every step. Yes.
It would. You know, you might consider it that in an organism if you need a taller one, but that doesn't explain complex arrangements of different types of tissue that provide functionality. That's what evolution would have to do a bajillion times over and over to produce all the body plans of all the living things in this world.
And yet, whenever anybody has given
What is
an
the
example,
scientific reason for believing that mutations, accruing mutations, develop information that produces something functional that didn't exist before?
because it's already functional. Everything is
Something
already
that didn't
functional.
exist before.
Yeah, it already exists as something, and that something it exists as is already functional.
What is the evidence that genetic mutations build functional new information?
The fact that we know that mutations change things and that these changes can be beneficial to certain organisms and certain environments.
Where are these beneficial mutations?
Change in femoral length. Good God, man.
So where is the reason for believing that changing the length of a bone is sufficient to explain the arrangement of different types of tissues
Because it is an
in a
example
functional
of
manner?
a change in the size, shape, relative proportion, relative
Of a
protein expression of a tissue,
of a
which is
bone.
exactly the mechanism required by evolution.
No, that wouldn't explain the coming into being of an eye or the stinger in the tail of a scorpion.
If
That's
you apply the same
numerous
mechanism,
tissues.
if you apply the same mechanism out, it can account for the eye and the stinger of the scorpion as well.
No, because the keratin's already not there. We've been over this a long time.
Keratin is a change in protein expression.
You're going in circles because you can't answer the question. I have to take a phone call.
I'm going in circles because I'm answering the same question I've already answered. Anyway, Standing, I sort of agree with you that I think it's now 2 o'clock and this has gone on much longer than I thought it would. And I told you it would be a train wreck.
You know what, guys? I appreciate having you guys on. It was still fun nonetheless. Yeah, it's 3 a.m. here now, but
yeah,
I think we'll end it here.
We do not get along with one another.
It's a love-hate relationship.
Yeah, I wouldn't even say that.
Well, regardless, it was fun. Let's all get some sleep. The phone ringing might have been a sign to end it. Athena passed out a long time ago.
It was the sign that apparently he was so loud that his neighbors are calling him.
Are you debating again?
All right.
Okay.
Sorry your originally planned hangout didn't work out. Glad I could fill in on short notice. And we'll call it a night there.
Sounds good. You know what? I'm sure Immutable Destiny will thank you as well. But thanks for filling in. We'll talk to you guys later. Have a good night.